Nunez. I'm Lia Nunez Casillas from the Gota group in the University of La Laguna, together with Floren Mican, Patricia Tito, Manuel Arbelo, and Ben Sommers from the hyperspectral group in Vigo, Belgium, also took part in this work entitled Plant Species Monitoring in the Canary Islands using WB2 imagery. So during this presentation, I will first introduce our object of study, which would be the laurel forest in Canary Islands, also known as laurisilva, and then about the WB2 data and the technique we chose for trying to map the biodiversity and the species composition of this forest, which is a spectral mixture analysis, particularly MESMA technique. Then I will go to the data collection and pre-processing phase, which turned out to be one of the most delicate parts of the study. And then I will talk about the implementation of the spectral libraries and members that are needed to run the MESMA analysis. These spectral libraries, as I will explain later, were composed by a field spectrum with a spectral radiometer, a hyperspectral radiometer, and straightforward from pure pixels in the WB2 image. Finally, I will talk about the mixing itself and the present work, because I will present the preliminary results we had in the moment we published this work, but I will talk a little bit about the future work we intend to do to improve this analysis. So this laurisilva would be we would call technically endemic Macaronesian laurel forests and heathlands, which are located in this in the Atlantic, are a set of volcanic islands, and within the bioregion of Macaronesia. These are the Canary Islands, and marked in red are the 4 islands where the 5 islands, sorry, where the laurisilva is present. And precisely this summer there were, maybe you saw it in the news, very huge fires, forest fires, especially in the smallest island of La Gomera. And these forests are very endemic and very old. These are very relictual. So it's very difficult to recover the potential area distribution of this forest. So these are reasons why an improvement of the monitoring techniques of these forests would be interesting. So in Tenerife Island, when we performed the study, it's only at 10% of the potential distribution area at present, and which would be the dotted area here. The black areas would be the laurel forest and heathlands, and the well-conserved laurel forest would be reduced to these little areas. And another problem is it's only constrained to the northeast of the islands. So this is our study area. This is the aspect of the laurel forest, and for those who intend to map forest, you will notice that this is impossible because it's a very dense canopy, it's a very complex vertical structure of the forest, species overlap. Sometimes it's not so easy to identify local maxima, for example from LiDAR data. And there's a richness of species, but all the species have converged to very similar features of the leaves. So it makes it much more difficult. There's also a spatial heterogeneity linked to the heterogeneity of the landscape and the topography itself of the island. And there's a system of microclimates. So we will have to study at first at a very small scale, which we did. So the hierarchical measurement, just an introduction, is for those who don't use spectral mixture analysis. It's a special technique, a classification technique, where the output would be a cover fraction for each pixel of the end members or pure spectra previously given to the system. So if we want to know which proportion of each of three species there is in one pixel, we will use this spectral mixture analysis. And in our case, as we have a set like about 10 species, different species, we will do consecutive MESMA cycles. The MESMA stands for multiple and member spectral mixture analysis. The particularity of this technique, developed by Darrell Roberts in California, is that we don't have a unique end member or reference spectra for a species, but we have a whole spectral library for each species. So the spectrum, the unmixing technique, will choose the best fit spectra for each pixel. So in this study area, we had a pine plantation. Together with the fires are one of the reasons why these ecosystems are at present quite fragile. And there was a degraded area, which were former pine plantations, and they're actually recovering very slowly. And then we had nature forest. And those are the species we started the dominant species in that area we started to study. Eucalyptus globulus is also present there. And Morella faya, Laurus novocanariensis, and Ilex canariensis, we were able to collect pure pixels and field spectrum, both of them. For the others, we could only take one of them. So, in summary, this would be the different data we used and the aim of each data set. The LiDAR were used to auto-rectify the worldview to image, and also to get a digital surface model to make it easier to identify crowns. Then this was helped as well by the panchromatic band of worldview 2, which has a better spatial resolution, even though we couldn't use it for the spectral mixture analysis. It was very useful for the crown delineation. And the worldview 2 multispectral data, which are composed by 8 different spectral bands, 2 in the near infrared, 1 in the red edge, and it has additional bands like the yellow band and the coastal blue. This was used for the pure pixel and member collection and for the unmixing. Then we also use GPS data for pure pixel validation and the ASD field spec data for member collection directly from the field. So in the mature forest we needed a crane because we couldn't use the spectra from the leaves in the shade, which are have very different spectral features from the top of the canopy. We found that we had to split and sometimes spectra from the same species because they were in different phenological states. These are two examples. So Erica arborea with or without flowers have very different spectral characteristics, or with or without fruits. Then for the pure pixels selection in WorldView 2, we used a pan-sharpened WorldView 2 image for better visualization. This is, well, and on the left we have the original 2-meter resolution WorldView 2 image. On the right we have the pan-sharpened image. And we also use the digital surface model to detect the most remarkable crowns. Also, we didn't have such a good antenna as the previous presenter, so we had to go for very clear reference points. And from there, we had to reproduce a plot, a grid of 2 meters, and then find which rows were in the middle of the pixel. And we found that some band combinations of WorldView 2 were very useful to differentiate between different spectral groups of vegetation. For example, the yellow band, and some vegetation indices were very useful to tell between some species. For example, Eucalyptus globulus was very clear with the anthocyanin index. In some cases, we needed to do an empirical intercalibration between the hyperspectral field measurements and the pure and the WorldView 2 spectrum. We did it with the Morella faya, which were the most the species where we had the more information. And these are the spectra used, the spectral libraries for each species used for the MESMA. So, at the MESMA, the mixing, what we did was to compute a separability index, which would an interclass separability index, which would account for the variability between different classes or species, with respect to the variability, the inner variability within the species, the class. So this made in an iterative process. What we did was to merge very similar spectral libraries at the first cycles of the MESMA, and then gradually splitting them until we had all the species separately. One is this. As an example, this would be a result for Morella faya, where here we can see these are proportions of the species present in each pixel. Pixels with bright yellow have a proportion close to 1, 100% of the fraction cover. I put this example because it fit quite well with the field information we had. In other cases, it wasn't so easy. For example, we had to this RGB combination. Here we had to merge Mirica faya and Laurus novocanariensis in the same spectral library because they had very similar features, and I represented in red. In the second MESMA, we split Morella faya and Laurus novocanariensis, and now we see only in red Morella faya and in green Laurus novocanariensis. These are the overall results of these fractional covers we got for the different vegetation covers in the mature forest, the degraded forest, the pine plantation. In pine, it didn't work so well, and the rest, it was quite satisfying. Then, in the table 2, we have the distribution in terms of overall area, accounting for the proportion of each species in the pixels. At present, we are working in a more rich intercalibrated spectral libraries of N members. we intend to add a temporal factor in this MESMA, where we could choose the season which would be better to split to tell between different species according to the phenological phase. And also we are refining this hierarchical MESMA, and we intend to do a weighted MESMA, which each wavelength will have a weight proportionally to the separability power of the wavelength. And after that, we want to include the MESMA results in an object-based classification with LiDAR data as well. And that would be all. Thank you. Oh yeah, they do, especially because the laurel forest tends to grow in very shady places. So another thing we want to do is to that would be in the front delineation phase, to select pure pixels. We want to model the shades, comparing the lighter derived shade map and with the panchromatic band as well, so that we and also the MESMA gives us an alternative, which would be a shade map, and it's derived from the MESMA can tell like attenuated end members, and can estimate, it gives an estimate of the shades per pixel, the shade proportion per pixel. And we want to compare these different results to account for the shade. Another thing would be to characterize the proportion of shade present in each vegetation cover as another feature for each vegetation type. Because it's much more easier. The problem is that sometimes there's no pure pixel. So we needed field measurements which add like intercalibration problems between the satellite and the field. But we could manage somehow. So then we have to